Welcome back to the Seized Drugs and Toxicology session of our symposium. For this half of the session, we'll turn our attention to novel ways of identifying drugs, particularly novel psychoactive substances. Our first presentation for this portion of the session is from Dr. Edward Cisco, a research chemist in the Surface and Trace Chemical Analysis Group at NIST, and Dr. Arun Murthy, a mathematical statistician in the Mass Spectrometry Data Center at NIST. They will discuss the development of an open source DART search software and library building tool for the analysis of complex drug mixtures. This work was also funded under the Public Labs program and resulted in the publicly available and practical tool. Welcome, Dr. Cisco and Dr. Murthy.
Awesome. Well, thank you. Hello, welcome to our discussion of the development of an open source direct analysis and real-time mass spectrometry or DART mass spec search software and library building tool for the analysis of complex drugs. I am Dr. Arun Murthy, a mathematical statistician at the National Institute of Standards and Technology, and I will be co-presenting with my collaborator and friend, Dr. Ed Cisco, a research chemist also at NIST. So just a few quick disclaimers that you can read. Importantly, NIST does not necessarily endorse or recommend any of the commercial products that we mentioned. And a portion of this work, as you know, is supported by an NIJ award. So what is this talk all about? Most scientific research or most software that evolves from scientific research is perfectly functional, but usually unusable. And this is because as scientists, we're usually trying to write code to deal with a certain specific challenge and, eventually really just explore a research area. And so sometimes our functional research doesn't actually get to the hands of people that might use it in practice. And so that was kind of our goal with this whole project with this NIJ funding, was to take our functional research and build a usable tool. So a little background about the functional research. Seized drug analysis is the most commonly requested forensics examination or examination in the United States. Forensics labs are swamped with challenging case samples, and this has been the case for several years now. And so an appealing technology for these forensics labs is ambient ionization mass spectrometry. One example of ambient ionization mass spectrometry is direct analysis in real time paired with a high-res mass spec or DART mass spec. The appealing thing about this is there's a lack of chromatography, which makes taking these measurements incredibly rapid. The challenge is that the lack of chromatography results in incredibly complex data. And so it's not ideal for most mass spec searching or interpretation tools. And so naturally, there are two things that we thought we could help with given our experience. One, we could construct or put together a NIST DART mass spec forensic database. And so in late 2020, we put together an updated version of the DART mass spec database, which at the time contained about 600 in-source collision-induced dissociation mass spectra, or ISCID mass spectra, measured at multiple fragmentation levels. You can see this pie chart here. We had several important compounds that were of importance at the time and still currently important to the forensic community. The current version, which we call Earthworm, was just released, and it contains 943 compounds. And the library is available as both NIST format to be used with NIST MS Search, or also as an R data table to be used with the NIST NIJ data interpretation tool that we'll be talking about today. So most people use pure compound libraries to identify pure compounds. The thing with the DART mass spec, and especially in its application in the forensics community, is that the DART mass spec is used for pre-screening. Pre-screening seized evidence that may often be mixtures. And so your traditional mass spectral library search algorithms, of which there are many, that leverage these pure compound libraries aren't actually useful for that use case of mixture identification. And so we realized that we have to address this if we want people to be able to use our library effectively. And so one afternoon, or actually several afternoons, Ed came by my office. We worked at the blackboard. We talked a little math, we talked a little chemistry, and we came up with this new method for identifying mixture components I won't go into the details of the algorithm, but other than say its name, it's the Inverted Library Search Algorithm, or ILSA for short. We implemented the ILSA as a simple application. We tested it with several laboratory mixtures. We made the source code publicly available through GitHub, and you can see that link here. And we wrote a paper summarizing the algorithm, summarizing what we learned, the computational research trifecta: software, solve a problem, write some software, write a paper. And so we thought we were done. But of course, and again, this is the whole point of this talk, is that the ILSA, as useful as it was to us, we knew it would be even more useful to practicing forensic scientists that were really doing drug screening or doing DART mass spec in forensics labs. And so to speak on the process of converting our functional research into a usable tool, I'm going to pass it over to Ed.
Thank you. So as Arun said, the focus of this talk is how do we take that functional research and bring it into a usable tool. And so as you can see on the right-hand side, we have our functional research, which was our spectral library, the ILSA, other expertise Arun has in mass spec tool development. And what we realized off the get-go was in order to develop this into a usable tool, we need to make sure we have end users involved in that development process. So with the help of the funding from NIJ, and then along with 5 collaborating laboratories that you can see on the left-hand side here, we worked to iteratively develop what we call the data interpretation tool, or the DIT, which is down there in the implementation piece. And so our collaborators were crucial in going through and providing feedback on what functionalities work, what don't work, what things are missing, how do we get to where a tool that they would use at the end of the day in their labs for actually making reports, identifications, and things like that. So to walk you through that kind of development process, we have a timeline to show you. So the development of the DIT started back in October of 2020, where we did, as the functional research component, like Arun said, we published the ILSA. We established some initial functionality of the DIT and released the first DART mass spec database. Around December of 2020, we shared the very first version of the DIT with our user group to get some very initial feedback on what are the major things we need to include, not include, things of that nature. And at that time, we were also working to bring on someone with software development expertise to really help with the usability and functionality of that piece. And we also continued along with a roughly quarterly updates of our database. After giving it to our user group for a few months to try out and play with, we went through and wanted to collect some feedback. This was done through a number of different mechanisms, just communications verbally and through email, but also through a tool survey. And so the first survey we sent out, one of the big things we heard uniformly was reporting is a big component of what we need to be able to do with this tool that we're developing. And so there was a lot of questions as to, in order to do reporting, what do you need to have included? What fields do we need to be capturing, whether that's case numbers, instrument IDs, dates of analysis, and then what other functionalities are critical in that development process as well? What other things will help you answer the questions at the end of the day with seized drug screening functionalities that need to be included in that? And so we took that results from those surveys, went back to the drawing board, worked on a second version of the DIT internally. By this point, we had a software developer with us as well, helping to develop kind of the overall experience of the program for everybody. And again, a few months later, we went back to our user group, shared the updated version that had things like reporting, different scoring metrics, different settings you could choose and change, the ability to load multiple mass spectra into a single search. And then again, we went through and collected user feedback. Since we were more focused now on getting towards a public release of our target of October of 2021, our second survey was really focused on reporting bugs. And so were there any major issues that need to be addressed? B, were there any bugs that came across in the software that we need to address prior to public release? And then C, we developed a user manual, a short user manual to go along with the software. We wanted to also capture what was the utility of that user manual. Is there anything that needed to be included, wasn't clear, and things of that nature. And so we targeted a October 21st release of the public version 1 of the DIT, which we did. So again, after that second survey, we went back, made some very minor tweaks. And then a few weeks prior to the public release, we shared our public release version of the DIT with our user group, along with the most updated DART mass spec database at that point. shared that, made sure everything was good to go. And then on October 21, we publicly released the software through an FTCOE webinar. Since then, we've continued to work on the project. We just released a few weeks ago version 1.1 that has some minor tweaks to scoring to help improve scoring, and then also coloring targets versus, which we'll show you in the next slide, colored targets for things that we can identify versus what we can't identify in the database. And then finally, we have the next obvious update, which Arun talked about earlier, Earthworm for the DART mass spec database. So you may be asking how this all works and what this looks like. Here's a quick little demo for you. So this is the layout of the DIT. A user would go in and upload multiple in-source CID fragmentation spectra of a sample. In this case, we're uploading both a low, medium, and high fragmentation spectrum. It's as simple as clicking search database, and you will see your mass spectrum. In this case, your low fragmentation mass spectrum pop up, the identification of the possible targets, along with the matches for each of the targets. And we have two different scoring metrics for those matches. And you can see there are things that we identify that don't have hits in the library. We can actually go to the settings. We've tried to minimize settings as much as possible, but one of those is to only show things where we can match, which is what we're doing here. So now we can go through and you see we have different scores. So depending on how the ILSA, the comparison of these 30, 60, 90 or low, medium, and high fragmentation spectra between the database and the query look, that's what's contributing to your scores there. Like I said, reporting is a big functionality, so you can enter in this metadata up top and then simply clicking a reporting feature and you can generate a printable or PDFable report that outlays the metadata, your spectra, and the results of the analysis. You can also go through and search the library that you're using to do the analysis if you're interested. So in this case, we can go through and search for cocaine. And again, you'll see all of the metadata that we have collected with it, molecular mass, base peak, things like that, as well as the 3 different in-source CID fragmentation spectra, your low, mid, and high fragmentation. And just like with the search part of that software, you can also pull up a report version of the library spectra if you want to be able to print those out for comparison purposes or things like that. So if you're interested in playing with the DIT or downloading it to use it, here's how you would go about doing that. It's freely available on the NIST public data repository, either through the DOI down below or the QR code presented here. It's just a matter of downloading the zip file, extracting it on your desktop, and then opening the folder, hitting run, and you're off to the races and being able to use it. The DIT is vendor agnostic, so it is able to accept a range of different file types, also a range of different mass tolerances. So you can go anywhere from integer mass up to high resolution mass spec measurements. And we do have data conversion tools for certain software vendors as well that are included in that DIT. And then also in that folder is the quick couple-page user manual that really just provides people how to open the software and run it and get some initial results. If you have more in-depth questions, we're always around to answer. And if you're not able to download stuff from the DIT, you can also email us at dartdata@nist.gov, and we'll be happy to mail you a USB dongle that has the program along with it. And I should also mention that when you download the DIT, you do get a number of example spectra to play with too. So if you don't currently have DART or any other amp ionization tool, but you're interested in seeing how this works, we do include a bunch of different example spectrum for you to play with. And so to wrap things up, even though we are, the NIJ funding has completed, we're still continuing. We see the utility of this software as by the community and the feedback that we've been getting. So we're continuing to develop additional features and functionalities, both on the software side and more on the R&D side. And so some of the things we're working on the software side is improved formatting, which you can see on the right-hand side here. This is kind of our new view of when you do a search. Everything's now in sortable tables to simplify being able to read through and compare results from within targets, outside of targets. We're also looking at things like adding in isotope measurements or identifying major fragment ions for people. Some other more software related things is currently right now we only support positive ionization mode in version 1.1. Version 2 will be able to allow you to do searching and have libraries for negative ionization mode as well. And then down the road, we're looking at implementing things like being able to search a batch of samples and for users to actually build their own libraries. On the R&D side of the house, we are working heavily on ensuring that this is not just a DART mass spec specific tool, and it actually is applicable to any sort of ambient ionization mass spectrometry. So we're looking at making sure that is true, seeing if you can use DART mass spec databases for other types of ambient ionization tools as well. And then really moving towards being able to develop tools to help identify unknown components in mixtures, and moving towards better scoring or other approaches to either add or increase confidence or provide you a more objective measure of confidence for the identification of compounds within your mixture.
Awesome. And so just to close the talk, we wanted to share one of these incredible comments that we got from one of our collaborating labs, the practitioner Sarah Kern at the FDA Forensic Chemistry Center. This entire process has been incredibly enjoyable and rewarding. And it turns out it was also rewarding for the labs that were participating with us. And so, yeah, I guess we're super thankful to the NIJ for the funding, which allowed us to, participate in the type of research or the type of project that we don't normally do. And it was really rewarding, and we are quite grateful. So thank you for your time. And if you have any comments or questions, feel free to let us know at the email listed here, dartdata@nist.gov. Thanks.
Thank you, Ed and Arun. Audience, please continue adding your questions to the chat for Dr. Cisco and Dr. Murthy to answer during the Q&A portion. Continuing with our DART theme, our next presenter, Dr. Rabi Ann Musa, will discuss the fusion of DART high-res mass spec-derived dark matter and IR spectroscopy for the identification of new psychoactive substances. Dr. Musa is a professor in the Department of Chemistry at the State University of New York at Albany. Thank you for being virtually here, Dr. Musa.
Hello, everyone. My name is Rabi Musa, and I'm a professor in the Chemistry Department at the State University of New York at Albany. And I'm going to be speaking with you today about how we are fusing DART mass spectral data and infrared spectroscopy data to aid in the identification of new psychoactive substances. So I'm going to begin with a disclosure. This project was funded in part by the National Institute of Justice, but the opinions, findings, and conclusions or recommendations that I express are those of me in my lab and the lab members, and not necessarily those of the Department of Justice. So I'm going to begin with an overview, with a background of how we got into this project and why we're doing it, the two instrumental techniques that we use, which are DART mass spectrometry and infrared spectroscopy. I will talk about how we apply multivariate statistical analysis to the output of those methods, and then how we apply the outcomes of that to structure determination of unknowns, and then I'll wrap up with some conclusions. So the primary goal of the work was to address some of the unique structural elucidation challenges that crime labs are experiencing because of the emergence of new psychoactive substances, and coming up with a way to rapidly address that issue by crime labs. Now, there are broad ranges of these types of substances or classes. There are cathinones, opioids, tryptamines, and cannabinoids. I'm going to be focusing today on cathinones and tryptamines. Now, the type of challenge that a crime lab typically experiences is exemplified by the report here in this publication, which was in Forensic Toxicology in 2016, published by Kaizaki and others. I'm going to walk you through the experience that they had. They basically had submitted to their crime lab numerous samples of new psychoactive substances, an EI mass spectrum derived from a GC MS experiment is shown here for an unknown. It's pretty nondescript with one base peak. And it turns out that if you screen this against the NIST database, for example, you see multiple mass spectral hits that have EI mass spectra that are very similar to that of the unknown. And the researchers knew that it couldn't be any one of these compounds because the GC retention time for this unknown was quite a bit different. And so essentially the problem you have is a missing molecular ion peak, very few fragments that you can use for structural determination. This particular class of compounds, namely cathinones, often fragment so extensively by EI that there is very little useful structural information that can be gleaned. And so you also have many molecules that have a very similar fragmentation pattern. And so essentially when they found no matches in the NIST database or in the Cayman library, they ended up acquiring high resolution mass spec data, which allowed them to get molecular formula information. And then they had enough compound in purified form so that they were able to do 1 and 2D NMR experiments. And through that, they were able to determine that this is the structure of that molecule here in the upper left-hand corner. And you can see that it is an isomer of these other three compounds. And so what you can also see is that if you were to look at these compounds and analyze them by infrared spectroscopy, you would essentially see in the functional group information segments of the spectra, the same sorts of functional groups, basically the same peaks, but perhaps in the fingerprint region, because of the differences in structure around the exocyclic green nitrogens, you might be able to detect that these compounds are different. Now, another case, this one is a tryptamine. These are tryptamines we're looking at here. You run into a similar problem. So here you have two isomers. They differ, again, in terms of structural features at the exocyclic ring nitrogen, but their EI mass spectra are very similar. And in fact, they have very, very similar GC retention times, which means that if this came into evidence in the crime lab and these structures were unknown, it would be very, very difficult to know what they are. And so the question we had was whether there might be an alternative MS approach that would yield more interpretable spectra that could be used, whether or not IR spectroscopy could be integrated into the workflow in a seamless way to further inform spectral interpretation. We wanted to know also whether there were chemometric tools that could be used to inform structure determination, and whether all of these approaches could be harmonized to determine the structure. And so we showed in previous work that you could actually do this for cathinones. And so the student who worked on this, who has since graduated and gotten her PhD, Kristen Falbel, was the lead on this project. And we showed how you could do this for cathinone unknowns by applying statistical analysis processing to DART mass spectral data, but specifically what we're calling neutral loss spectra. And I'm going to walk you through that process to show you how it goes, and then show you how we applied it to tryptamines. So we used DART mass spectrometry, but specifically high resolution mass spectrometry using the JOL AccuTOF system. It's nice because we can perform experiments under soft ionization conditions where we see little to no fragmentation, but we can also increase the voltages of the experiment up to 90 volts so that we can get lots of fragments that can then be used for interpretation. We have millimass unit accuracy with this mass analyzer and nanogram detection limits. And there is little to no sample prep. In other words, if you have a purified substance like a solid, you can analyze it directly and you don't have to do any extractions. So to show you how this works, we're going to start with looking at these two cathinones here, and they differ from one another in that the bottom one has an additional CH2 group. So that's the MDP-BP versus the MDP-PP. And if you look at the EI mass spectra of both of these, you can see that they're pretty nondescript, and you don't have very much in the way of fragments that you can work with, and the molecular ion in each case is missing. But if, on the other hand, you look at the 20-volt spectra obtained by DART, you get these wonderful high-resolution masses from which you can extract molecular formula information. And furthermore, if you were, for example, to analyze these two compounds by DART HRMS under collision-induced association conditions, so that would be 90 volts in each case, you have a range of fragments that you can work with. And again, this is not something that we saw when we were looking at the EI mass spectra. Now, one of the things that you can do with these mass spectra that are collected at 90 volts is you can take advantage of the fact that the protonated precursor is still visible in this spectrum. And so if you were, for example, to subtract the high resolution masses of the fragments that you get from the protonated precursor, you can get the masses of the neutral fragments that are lost, or the dark matter of the spectra. And so you can actually see that here. Each one of these, when you do that subtraction, you can actually get this virtual neutral loss spectrum where you have high resolution masses associated with fragments that are lost from the protonated precursor. And so each one of these peaks that you see in this virtual neutral loss spectrum is associated with a fragment. And so, for example, the pyrrolidine system here is associated with the mass 71. The one at 99 is associated with the pyrrolidine plus the alpha carbon and the substituent attached to that, and so on. And so you can run through the structure and assign all of these neutral losses to segments of your molecule. Now you can do the same thing for the second structure that had the additional CH2 group here, and you can go through that and also use the software to readily generate a neutral loss spectrum like the one we have here. And Each of these peaks, again, will be correlated to some particular fragment in the molecule. And so you can just run through that and see that each one of these can be assigned, as I'm showing you here in this neutral loss spectrum and these color-coded boxes that you see in the structure. Now, if you were to compare the 90-volt spectra for both of these molecules, like we're doing in this mirror plot, where there's a spectrum in blue on top and a spectrum in red on the bottom, even though these compounds are highly structurally related, you won't be able to simply look at the patterns here and see that. However, if you were to instead create a mirror plot using the neutral loss spectra, you can actually see that some of these actually even look like mirror images of one another. And what this indicates is that you're getting identical neutral losses and that these molecules are fragmenting in a similar fashion. Now, what's nice about that is that you can actually literally take hundreds of these molecules, generate neutral loss spectra, and then subject those data to hierarchical clustering analysis. And if you were to do that, all of the compounds that have similar structural features and fragment by similar mechanisms, yielding similar neutral fragments, all cluster together. So for example, in the yellow arc of this dendrogram, which is the output of this analysis, you can see that all of the samples that are similar in that they have this pyrrolidine system here at the exocyclic ring nitrogen, well, exocyclic from the benzene ring, you can see that they cluster here, whereas, for example, those that have the pyrrolidine system incorporated into their structures appear primarily down here in the green arc. And what this means is that if you have an unknown and you can generate its neutral loss spectrum, you can screen it against the dendrogram, and depending upon where it falls, will tell you something about what the structure is likely to be or what its main features are. Now, when we did this for tryptamines, which I'm going to be reporting to you now, we added an additional dimension, which is to fuse the data acquired by DART HRMS at 60 volts as well as 90 volts. And so what you see here are cartoons of both types of spectra. They tend to share peaks, but they also have peaks that are distinct. And so if you take that data and you fuse it, you get a new virtual spectrum that has many more peaks that can be used as the basis of structure determination. So we did that. So here what you see is a scheme that shows you the data generation and the analysis workflow for the tryptamines. We generated DART HRMS data at 20, 60, and 90 volts. We computed the neutral loss spectra from those, and then we fused those, as I showed you on the previous slide. In addition to that, we collected IR data for all of the samples, just in case we needed it, to help refine our prediction capacity for the statistical analysis model that I'm going to be telling you about shortly. And we fused all of those data row-wise, imported them into MATLAB, and then analyzed the data there. So that's the first part indicated here, where we fused all the data. We first subjected it to hierarchical clustering analysis to check to see whether we would get clustering, which would mean that inherent in the data were the attributes that are necessary to enable us to get class distinctions. And then we subjected it, once we found out that we could do that, we subjected the data to a supervised learning approach. Specifically, we use partial least squares discriminant analysis, or PLSDA. So what you see here on this slide are the range of tryptamines that we looked at. And this slide serves to show you all the ways in which the core scaffold can be modified and still retain the psychoactivity of these compounds. So when we acquired the neutral loss data and subjected it to statistical analysis, specifically hierarchical clustering, we got this wonderful dendrogram, which is color-coded according to the 10 groups that emerged from this. And what this dendrogram and its color coding told us was that like things cluster together, which is fantastic. With this information in hand, we were able to apply PLSDA to the model, and we were able to assess its predictive capacity using 100 times bootstrap sampling. And so the merits of that, the outcome of that are shown in this table. And we were happy to see that in terms of sensitivity, specificity, and precision, we got 100% accuracy. So everything that it actually predicted, it predicted correctly. in terms of the identity of the molecules and the classes into which they fell, and the not assigned rate was 3%. So when it didn't know what it was, which was 3% of the time, it basically didn't try to classify it, which is good. Now, one of the nice things about using PLSDA is it allows us to generate these variable importance and projection scores that give us an indication of just how important a particular variable, or in this case, M over Z value, is to classifying a particular molecule. And that's nice because it gives us markers for the different groups. And so the variable importance and projection, or VIP scores, are indicated here on the left panels for the 60 and 90 volt data. The VIP scores extend from 1 to 9, which you can see on the y-axis, and on the x-axis down here, and on the y-axis on the left-hand side, you can see the M over Z values for the 60 and 90 volt data. So essentially what this is telling us, for example, for 60 volt data, is that if you have M over Z 161, it has a very high VIP score, which means it is heavily weighted in enabling discrimination between classes. And it happens, if you look at this table, to be very important in distinguishing group 5 from other classes. And so the M over Z values that you see in this table are for the discrimination of the different classes that you see here. So for example, for group 10, 189 is an important mass, and for group 7, 147 is important. So the question we had then was whether this model could be used to identify new tryptamine unknowns that were unfamiliar to the model. And so for that, we did some external validation, looking at 4 different structures, which are depicted here, and their nicknames are also shown. For example, 4-acetoxy malt, where you have an acetoxy group here on the benzene ring, and the 4-hydroxy malt, where you have a hydroxy group here on the benzene ring. So we generated the neutral loss spectra for these, and when we screened them against the dendrogram, we discovered that the 4-hydroxy malt fell in the red segment of the dendrogram, where indicating that it was in group 7, and the other 3 fell within the group 10 area, which was green. And what's great about that is that that's exactly where they should have fallen. And so the probability of prediction is shown here. And so it ranged from 70 to 100%, but in all cases, the prediction was actually correct. Now, if you go back and you look at these spectra, you can actually see that contained within these neutral loss spectra are markers that you would expect to see. So 189 is a class 10 marker, for example, and 147 is a class 7 marker. Now, what are these markers associated with? Well, if you look here, for example, at the 4-acetoxy malt, that peak at 189 is associated with this fragment that you see in the red box. And for the 4-hydroxy malt, the peak at 147 is associated with what you see here in this red box. So how would a person in the crime lab go about using this scheme to figure out what sort of structure they have or what class of molecule they have? Well, it turns out, let's try to do that for 4-hydroxy malt. So here is your molecule. Here's your high resolution mass. From that, you can extract this formula. And because the mass is for the protonated precursor, you need to subtract a proton, a hydrogen, which gives you a formula of C14H18 and N2O. And since we know what 147 in its spectrum is attributable to in its neutral loss spectrum, we know that we can subtract C9H8NO, which is what this is associated with from this formula. That leaves us with a balance of C5H10N. And that formula indicates that we have 1 degree of unsaturation, which basically means that our structure has to have this core scaffold. And the only distinction that you see then is with what's here attached to the exocyclic ring nitrogen. And so it's got to be one of these 4. And it turns out that it happens to be the one here in red. Now, just to illustrate how powerful this is, if you were to take that initial formula and screen it in SciFinder, you would get 32,000 possible molecules. Whereas if you were to take that structure and put it in this database, you would come up with 28. And even though that's a lot smaller number, none of them happen to be, at the timing of this presentation, the correct structure. And yet doing it in this simplified way allows you to predict that it's one of these 7 and readily figure out that it happens to be this one from additional work that you could do. And so the question then becomes, what happens when the DART HRMS data are insufficient to enable a single group classification? An example would be for 4-hydroxy-NET, which the sample gave, which the model gave an assignment to group 1 as well as group 4. And this is where infrared spectroscopy can help you. So we actually collected IR spectra for all of these samples, also subjected that data to hierarchical clustering analysis to get this wonderful color-coded dendrogram, from which emerged 7 groups. And you can see here from application of PLS-DA to that data with 100 times bootstrap sampling, that you get sensitivity, specificity, and precision values, which are very impressive, accuracy of 100%, but the not assigned rate is 10%, which is quite a bit higher than we saw looking just at the DART data. We were also able to look at VIP scores for the IR data analysis. Those scores that you see above the red line indicate that these wave numbers are important for classification. So it'd be nice in the future to go back and look to see what we can attribute those to. But more importantly, if you take the IR data and the DART data and you fuse them, you get a new dendrogram, which is even more accurate in its prediction capacity. So 5 groups emerged from that in terms of the merits of PLS-DA, using again 100 times bootstrap sampling. We had an accuracy of 100%, and the not assigned rate was 0, and the error rate was 0, which is fantastic. So now the final question is whether this model can be used to identify new tryptamine unknowns that are unfamiliar to the model. And so what we did was we analyzed several samples comparing just using DART versus IR versus a fusion of the 2 methods. And just to give you an example, if you just use IR, the accuracy is 100%, but the multi-label assignment is 30%. Whereas for the fused data, the accuracy is 100%, but the multi-label assignment drops to 3%. And so you can see that the fused model is far more robust and accurate than using DART and IR alone. And so based on that, we can conclude that you can use DART-MS and IR collectively to rapidly screen for new psychoactive substance tryptamines, as well as cathinones, and get readily interpretable fragment peaks. And that data interpretation can be greatly facilitated by using multivariate statistical analysis. So future work involves us expanding our databases with more new psychoactive substances. And hopefully this is something we can roll out that crime labs can use readily. So I want to acknowledge the folks who worked on this project. Dr. Behrami Sultan did the statistical analysis, and Monica Ventura did all of the DART analyses. I want to thank the National Institute of Justice for funding this work. And I want to thank you for your kind attention. I'd be happy to respond to any questions you may have.
Thank you, Dr. Musa. Next, we will hear from Dr. Glenn Jackson, the Ming Hsieh Distinguished Professor of Forensic and Investigative Science at West Virginia University. Dr. Jackson will discuss his work on the Expert Algorithm for Substance Identification, or EASY, for structural characterization of emerging synthetic drugs. Welcome, Dr. Jackson.
Thank you for the opportunity to speak today. I will start with the disclaimer that the opinions I'm about to present are those of myself and my group and not necessarily those of NIJ, who sponsored the research. This particular project, the development of a new algorithm for mass spectral identification, addresses 2 important needs of the National Institute of Justice. One is the ability to identify compounds by comparison to spectra that were collected on different instruments, and the other is to provide error rates and qualitative determinations. These are kind of long-standing problems in the area of seized drug identifications, and this is why many guidelines and recommendations that are out there basically say that when you try to identify a substance using, for example, GC mass spec, that you should analyze a reference standard on the same instrument using the same conditions. And the reason is that when you do that, you can minimize the random variance that occurs between replicate measurements and therefore have more similar looking spectra to increase the confidence in your results. That's the idea anyway. So what we're really talking about is one analysis method in an analytical scheme for the identification of a seized drug in this particular case. And so what we're talking about is GC mass spec, which has been around since the mid-1950s, and just showing in a simple schematic here is the separation on the GC, and then, sorry, I'm just trying to bring up a laser pointer here. Separation on a GC, and then ionization analysis by the mass spectrometer, and some kind of computer readout. And the results, most of you will be familiar with, look something like this. You separate the multi-component mixture as a function of time, and then at each moment in time, you can look at a mass spectrum and try and identify the substance based on those mass spectra. This is made easy when those spectra are not convoluted with one another, they don't overlap and things like that. So in the best case scenario, we might get something like this. We can look at a mass spectrum on a peak, for example, it might be the cocaine or suspected cocaine. We can send that to a database, and there's an algorithm then that compares the spectral similarity between that question or unknown sample and all the spectra in the database. And in this particular case, because we started with cocaine, the best match in the database is a self-identity. The second best match in the database is another replicate of cocaine. You can see that the third best hit in the database is pseudococaine, which is just a distereomer, just has slightly different stereochemistry, these two R groups up here. And you can see that the other distereomers are also quite good matches, and that's because they fragment really similarly to cocaine. Now, just as a kind of reminder, we always do have retention time to kind of work with to help us with our identities. And honestly, they can usually solve the problem in a case like this. But what I really want to do is talk about maximizing the power of mass spectral identifications, even in the absence of GC retention times. Of course, we can use GC as well, but I'm just trying to maximize the power in mass spec right now. So what we're doing is building on the work of others, the shoulders of giants, really. And so really since the 1960s, there has been a plethora of computer search algorithms, and some of the leading figures are shown here. And so we, in a way, our work is kind of building on that, because we're also developing a search algorithm. You know, these spectral identification of organic substances was really started in the 1930s when Liguo first developed the electron ionization source for mass spectrometry. We're going to be using principles like physical chemical principles of unimolecular fragmentation, which were first described in the 1920s by Rice, Ramsberger, and Castle, and then modified in the 1950s by Marcus to develop this more comprehensive theory. And finally, we're going to be using statistical techniques. It's just a simple multiple regression or multiple linear regression that was developed really by Galton in the late 1880s. And we're going to be really bringing all of this together in this one particular project. So let's look at the real source of the problem here. Here's just looking at cocaine across a GC peak, and I've zoomed in on the chromatogram. So we're just looking at about 8 spectra across one peak. And you can see on this dimension the mass to charge ratio, and in this dimension the abundance or the intensity. If we normalize to the base peak across this GC peak, you can now see that the base peak is always M/Z 82 in this case. The next most abundant is M/Z 182, and then the rest of the fragments down here. I'm just going to focus and zoom in on this particular region of the spectrum just to see what's going on here. And what you'll notice is that going across one GC peak, there are certain fragments which, even after normalization to the base peak, undergo some kind of systematic change. You can see that these particular two are increasing as a function of chromatographic timescale, and they correlate with one another. This is even after normalization to the base peak. There are 2 other fragments in here which are decreasing across the GC peak, as it were, as a function of time. And this is, again, after normalization. And there's other peaks in here which don't change, which means that they're basically correlating very, very well with the base peak. And so what we have is that even within spectra, or within replicate spectra, we have correlations between certain pairs of ions and anti-correlations between other pairs of ions, and no correlations between, for example, the green and the yellow. And, you know, this is part of kind of the awkwardness, in a way, or the variance that exists. And what we really don't want to do then is in our case, use this variance that exists to help make better identifications. Where does this variance come from? Well, in an electron ionization source, there are 3 main sources of variance. One is the differences in energy as you excite and form those ions and make them fragment. And this can be influenced, for example, by the acceleration potential of the electrons through the source, the internal energy of the ions before they're ionized, that could be the temperature, things like that. You also have variances in kind of observation time, which is really influenced by the kinetic energy of the ions as they're accelerated out of the source through the ion uptakes to the mass spec detector. And finally, you have deviations caused by changes in the intensity as a function of M over Z. This is basically spectral skewing or mass bias, and this is caused primarily by focus lens 2 here, which can focus or defocus ions of certain M over Zs. So for example, low mass ions can correlate together or high mass ions can correlate together. So basically, these are the potential sources of variance, and of course there are kind of others as well, but they're the main ones. So how those particular factors influence the kinetics of fragmentation is really what was thoroughly described by the RRKM theory. Now, what I've shown here is the Castle equation, which is kind of a precursor or kind of to RRKM theory, and there's three major terms. And you can think of the movement of an ion from its excited state to fragments, rather like trying to move from a large body of water like a lake through a gorge to another lake. And there are three main factors that influence the speed or the kinetics with which we can do that. One is the size of the lake, and kind of the bigger the molecule, the bigger the lake, the lower the probability of finding yourself at the junction of the gorge. Okay, that's the first one, and that's described by the degrees of freedom up here. The second is the actual ability to get through the gorge at all, and that's defined by the excitation energy or the activation energy. And of course, the lower the activation energy, the more probable it is that you can actually get through there. And the last one is called the tightness of the transition state, and this is the kind of a frequency factor. And this is described by the need for the molecule to actually orient itself in a particular orientation to be able to undergo the mechanism. And so a tight transition state would require that the atoms be in a very specific location to rearrange, and a loose transition state is one with a simple cleavage which can form. Another way to think about these loose and tight transition states is kind of like an energy landscape, if you will. So these loose transition states, there's very many more of them. There's lots of fast ways down the mountain, and these tight transition states require walking along a very narrow path, a very specific pathway, which of course is going to be slower, but there's many more directions you can go in ultimately. So when we look at our RKM theory, we can model a simple kind of hypothetical molecule, N+, which is made of A, B, C, and D, and that can fragment into primary fragments and then secondary fragments. And in this hypothetical case, we can make up, kind of the size of these particular fragments and the bond dissociation energies, and using our RKM theory and some other assumptions, we can kind of calculate some hypothetical rates for these different steps. So what I've shown here is relative dissociation rates for these different pathways at a specific energy and a specific time. And what we can do is change the internal energy or change the apparent observation time, and using these relatively simple equations for kinetics, we can kind of calculate how the kinetics change for each pathway as a function of internal energy or fragmentation time. And of course, each pathway changes independently. The mass spectrum you observe is kind of a convolution of all of those pathways. Okay. So what we can do then is basically look at the change in relative ion abundances as a function of time at a fixed energy. And when you do that, you see a kind of pattern that looks like this. And if we normalize, for example, to the base peak, as we would do in a mass spectrum, and the base peak in this case is always C+. So if we normalize to C+, or we plot relative to C+, the other ion abundances, you can see that whereas they were curved before normalization, after normalization, you get these linear trends, at least over a relatively short 30% difference in time, these really linear trends. So what this tells us is that we expect ion abundances to change, to not always be constant relative to the base peak, but to change. And those changes could be linear, sometimes positive correlations and sometimes negative correlations. Now, it's quite possible that one lab has a certain instrument vendor that has an ion source of a particular geometry and particular conditions that enable observation of fragmentation kinetics over a certain time period. And that can be shown by lab 1 here. And so the variance in mass spectra observed by lab 1 could be in this particular domain. And so the ion abundance of this particular fragment might be around 0.28 or 28% relative to the base peak. Lab 2 might have a different instrument or different operation conditions, and they might be observing their fragmentation kinetics over a slightly different time frame, which would mean that the abundance of this particular fragment after normalization will be something like 0.24. This is to say that the mean of lab 1 and the mean of lab 2 could be very different for a particular drug standard, but if you measure the variance within lab 1 and do a linear correlation, then you could actually extrapolate that linear plot to make predictions down here in lab 2. And that's the fundamental difference between this algorithm and pretty much all of the algorithms, is that we attempt to make measurements within one laboratory, assess that variance, and then extrapolate to other conditions outside of that to make predictions outside of that. We can also look at the variance in internal energy, and when we do that, we can see here that it also changes as a function of internal energy. This is at a fixed time, and again, there are linear relationships between ions and a particular base peak, and some of them are more linear than others. And again, it might be that the internal energy distribution of ions in lab 1 might be significantly different from lab 2, but as long as we measure and assess the variance, we can do a linear calibration and extrapolate to the other lab. So this is all very well and good, but is it true? So if we look actually at some correlations that exist, remember I showed you the GC peaks for cocaine and how they do correlate, at least across one GC peak. Well, we took 132 cocaine spectra from a crime laboratory and looked at the correlations that exist between all the different pairs of ions, at least the 20 most abundant for cocaine. And you can see that the second most abundant ion here, MOZ182, correlates really well with this particular fragment, R squared of 0.82. Again, this is after normalization to the base peak. You can see that the consensus or the average of all of these spectra is 70.2, and in the absence of any other information, this would be your best guess for what the abundance of 182 would be in an unknown or a kind of a question sample of cocaine. So this correlation that exists between these two particular fragments, you can see in this correlation matrix here for 182, 198. So the R is 0.91, the R squared is 0.82. And you can see that what I've shown in gold, the two most highly correlating ions in a particular row. And in general, the trend is that if you have a low mass ion, then the two ions that correlate most strongly with it are other low mass ions. The same with the middle mass ions, and then high mass ions tend to correlate with one another. So this is in general spectral skewing or mass bias. There are some exceptions. For example, MOZ77 and 105 are tightly correlated, and this is because the formation of 105 from cocaine simply uses CO2 to form 77. Sorry, it uses CO to form 77. So there's kind of a fragmentation pathway that leads from 105 to 77. So what we're going to use then, instead of this, this is a binary correlations, we're going to use a multiple linear modeling, which was really promulgated by Galton in the 1880s. One example of how to use multiple linear modeling is to predict the deviant height of a child based on the deviant height of the parents. And by deviant height, what I mean is how much taller than average or how much shorter than average. And what Galton showed, for example, is that if the mom and dad are both 4 inches taller than average, then the height of the child is going to be about 0.36 times 4 inches of the mother, plus 0.33 times 4 inches of the father. So it's going to be like 3 inches taller than average is what you would predict on average for a child. The reasons why are a little bit more complicated. It's regression to the mean with multiple variables. But still, what we're really looking at is a dependent variable that's predicted based on coefficients by other independent variables. And that's exactly what we're going to do. So again, here are the 132 mass spectra for cocaine. We've just looked at the abundance of 182 after it's been normalized to the base peak, which is usually 82. And the average for those 132 spectra in about 4 months is 70.2%. That would be both the consensus abundance. Here is the bivariate plot that I showed you before, 182 versus 198. And here is a multivariate. So this is 182 versus the predicted abundance. And this predicted abundance here's the equation that was used for these predictions. It's a linear combination of coefficients multiplied by measured abundances. So again, there's just 4 variables up here, and we're going to make these predictions. And you can see that now, of all of the variance that exists in the abundance of MOZ182 in those 132 spectra, 95% of that variance can be explained by this model. That's to say that the variance that exists in mass spectra is not random, it's explainable based on these correlations and RRK theory, presumably. So let's look at other pairs of peaks. Here's our M/Z94 versus the model for M/Z94. And you can see in this case, what's plotted in cyan is the training set. This is 132 cocaine spectra from one lab. What I've shown in this color down here is about 120 odd spectra from a second lab. We then have about a dozen or more cocaine spectra from the NIST database, and then some diastereomers. And you can see that in general, the cocaine spectra have smaller residuals to the regression line than the diastereomers or ecgonine methyl ester, which is shown in purple here. And again, this is just reminding you that Lab 1's variance is in this particular region, and for M/Z94 might have an average of about 50% after normalization to the base peak. Lab 2's data is all the way down here, and the average for M/Z94 is more like 32% after normalization to the base peak. And this is just a reminder, this is exactly what RRKM theory would predict, is that if you have different conditions or different instruments, that the variance that exists in one laboratory can be extrapolated to make predictions in another laboratory. We've done this for a wide variety of peaks, and you can see that in this case, the allopseudococaine for this particular peak has a larger residual to the line than any of the cocaine. In this particular case, allopseudococaine was quite well predicted for M/Z94. So what we're showing here is that we can have different models for different fragments in a particular database. And so for cocaine, we build 20 models, we can make 20 predictions. What do you do with those 20 predictions? Well, you have to somehow kind of combine them to get some kind of average error of prediction and see how well you do. So here's looking at a frequency distribution of predictions. Here we're looking at the mean absolute spectral residual, which is kind of like the absolute error for 20 predictions. This is using the consensus approach. So using, for example, 70.2 as your best guess for M/Z182. And when you do the consensus spectrum as your best guess for things, then you can see that all the replicates of ecgonine methyl ester and the diastereomers of cocaine, these are the known negatives, have a certain distribution like this, and the known positives, all the cocaine, that would include the training set and the test set, and the NIST spectra all have variances like this, or mean absolute residuals like that. And in this particular case, if you can imagine a binary classifier, like can you have a threshold that, so if the mean absolute residual is smaller than the threshold, you call it cocaine, and if the mean absolute residual is larger than the threshold, you call it not cocaine. Well, if you do that, you can see that if in this particular case, you might have 51 false negatives, these are all false negatives, and 3 false positives. And there's nowhere where you can choose a threshold that makes 100% accurate identifications. However, if we use our algorithm to make predictions, then you can see that all of the cocaine spectra, this includes the training set, the test set, and the validation set in this spectra, the mean absolute residual is always less than 3. And the mean absolute residual for all of the non-cocaine spectra, that's ecgonine methyl ester and all the diastereomers, is always greater than 3, which means you can have a threshold now that makes errorless determinations, at least for the data set that we've looked at. We can also look at other measures of spectral similarity or dissimilarity. In this case, we can look at the Pearson product moment correlation between those 20 predictions. And we can do that for the exemplar approach or the consensus approach. And you can see, again, there's no one threshold that makes errorless identifications. But down here, you can see that there would be a threshold of perhaps 0.998 that would make errorless identifications, at least for this data set. We've also extended the model to fentanyl and valerofentanyl, which are very hard to distinguish based on mass spectra alone. The retention times are a little bit different, which helps. But again, here's showing spectral similarity. We've looked at thousands of replicates of known negatives and known positives for these 2 compounds. And this is a data set that was provided by about 9 different crime laboratories. And you can see that now we do have some errors for our easy model. We can convert this binary classification and assess it with the receiver operator characteristic curve or ROC curve. And you can see that in our case, we make about 7% error rate for spectral identifications for valerofentanyl versus isovalerofentanyl, and that's without the GC chromatographic retention. So in summary, I just want to close by saying that we were basically able to demonstrate that quasi-equilibrium theory or RRKM theory predicts linear relationships between ions caused by random changes in instrument conditions. This general linear modeling enables one to extrapolate from data collected from one instrument to data collected in another instrument. And finally, the general linear modeling can resolve cocaine from its diastereomers or isomers like valerofentanyl from isovalerofentanyl. I just want to thank NIJ for their funding again. I want to thank the experts in chemometrics for their advice and help over the past year or two as we've been developing this algorithm, and just an acknowledgement to the students in my group who actually collect all the data and work up all the models. So thank you to them too. Thank you very much for your time, and I'll be more than happy to answer any questions.
Thank you, Dr. Jackson. Our final presenter for this session is Sarah Walton, a forensic toxicologist at the Center for Forensic Science, Research, and Education. Ms. Walton's presentation will center on novel psychoactive substances discovery toolkits. Welcome, Ms. Walton.
Hi, my name is Sarah Walton, and I'm a forensic toxicologist at the Center for Forensic Science, Research, and Education. And today I'm going to talk to you about NPS discovery toolkits, the real-time identification and dissemination of information regarding novel psychoactive substances. So I don't have any conflicts of interest to disclose. As I mentioned, I am a scientist employee of the Center for Forensic Science, Research, and Education. And this project is supported by the National Institute of Justice. However, the opinions, findings, and conclusions don't necessarily reflect those of the Department of Justice. So first, I want to talk a little bit about the world of NPS. So novel psychoactive substances are really just new and constantly evolving drugs. If we remember back to the beginning of the fentanyl analog epidemic, there were so many different fentanyl analogs coming out, whereas now there are more non-fentanyl related synthetic opioids. So the new synthetic opioid epidemic, where we have the U series compounds like U47700, or the AP series compounds like AP237 and 2-methyl-AP237, and more recently, the nitazene analogs like isotonitazene, metanitazene, and pyrrolidinoadenitazene. We're having the same thing with synthetic cannabinoids as well. Those synthetic cannabinoids that we're used to seeing, like MDMB, 4-ene-pinaca, and 5-fluoro-MDMB-pica, they're now starting to disappear with the class-wide ban that China had put out in July of 2021. Now we're starting to see these new age synthetic cannabinoids, a lot of which are unknown, but like the oxazid compounds like 5-fluoro-beziopoxazid or beziohexoxazid, and really just NPS generally, novel psychoactive substances are a continuous danger to public health and public safety. There are a lot of challenges with dealing with NPS. Really the biggest challenge is there's such a quick turnover of drugs. A drug could be prevalent on the drug market for a number of weeks or a number of months, but really quick turnover. And most labs or government laboratories have a really bad lack of time and the lack of funding to be able to keep up with all these new drugs that keep emerging. As we start to see more and more synthetic drugs come out, a lot of them are more potent than the ones that came before it. So we really have that need for sensitive instrumentation. So the NPS Discovery Initiative was launched in 2018. And this is an open access drug early warning system. So really just for the rapid identification of emerging NPS. And the goal of our NPS Discovery Initiative is really just to anticipate the drugs that are going to be emerging on the drug market. So there are 6 objectives to the NPS Discovery Initiative, with the first being NPS surveillance. So the goal of this is really just to either obtain or request to be synthesized new standard reference materials of any of the new NPS that might be emerging or that we think might emerge. And a lot of that we do with our partners at Cayman Chemical. And this is just to keep a comprehensive and up-to-date library database. So our library database, we hope to have really just a full scope of NPS. That way we can really see what the drug trends are like. And right now we have over 800 NPS in our library. The next objective is NPS monitoring. This has to do a lot with sample mining and data mining with our collaborators at NMS Labs. So sample mining would be real-time or future endeavors. So really just trying to see what is prevalent now, what will be prevalent, really just coming up in the future. And then data mining is more retrospective or past. So seeing the drug trends or when a drug first emerged. So the goal of NPS monitoring is to determine the newest drugs as well as drug trends. The next objective is NPS metabolism, and the goal of this is just to characterize the metabolic pathways of novel psychoactive substances. So just to determine the biomarkers in blood and urine and the differences between them, as well as identify if there are metabolites that have a certain effect or certain toxicity that may be causing other effects that we're not seeing because we're not actually monitoring these metabolites, or if they weren't in our scope. And what we do with this is we use in vivo analysis and then analysis by metabolite pilot. The next is NPS confirmation, and we do this with many different instrument systems. So this is really just the rapid development and validation of methods. So we want to establish that reference concentration data. That way, if any lab is also seeing the same things we are, we can say, you know, this is the concentrations that we've been seeing. This is the sensitivity that you might need to identify and quantify this compound. The fifth is NPS toolkits, and really this is just a consolidation of all of the previous objectives. And the toolkits have detailed information regarding a specific NPS, so all of the information that we have into one spot. And then finally, knowledge transfer, which is just the dissemination of this information to forensic science and criminal justice stakeholders. And we do this through our NPS Discovery website. We have a number of reports that we put out, as well as we have an email list, presentations, publications. Any way we can get the knowledge out, the better. So here I have a picture of all of our different reports that we put out at npsdiscovery.org, and just from left to right there. The first is new drug monograph. Then we put out trend reports. We put out trend reports for each class of drugs. So we have a trend report for each quarter, and then for each class. So we have one for NPS opioids, benzodiazepines, stimulants and hallucinogens, and synthetic cannabinoids, all separated. Public health alerts. And then we have our clinical reports with our clinical collaborators, the NPS Discovery Toolkit, our drug checking reports, and NPS scope recommendations. The one I really want to focus on today is NPS Discovery Toolkits. And really, this is just a one-stop resource. So this provides all the information that we know about one specific NPS in one document. And as I mentioned, this is an open access format to really just allow labs to be able to identify and quantify these new NPS without delay if they don't have the time and the resources to really keep up with the NPS that are constantly coming out. So the first NPS Discovery Toolkit that was released was for metanidazine, and this was released in July of 2021. And this consists of a lot of the reports that I mentioned earlier. So the public alert, the trend plots, the quarterly trend reports, a new drug monograph, analytical methods and concentrations, and metabolism. Okay, so the first report that we put out is the new drug monograph. So this is upon first identification. So this drug monograph for metanidazine was released in July of 2020. And this was for a seized drug powder that we received, and this was the first identification we had. So what this report includes is a general summary of metanidazine, as well as any information, any resources, any literature that we're able to find on metanidazine. Then it also gives the chemical formula, molecular weight, exact mass, as well as chromatographic data. So here, this isn't the full drug monograph. I just pulled out the chromatographic data for both GCMS and LC TOFMS. So you can see metanidazine in both up at the top in the GCMS data and at the bottom in the LC TOFMS, as well as our two internal standards. And then the drug monograph also includes our method parameters for both of these. That way that any lab would be able to reproduce it if they had the same instrumentation. The next piece of information that comes out is the public alert. So the public alert for any compound will have just a background and then a summary on the drug of interest. So this one, you can see it says metanidazine begins proliferation as the newest synthetic opioid among the latest cycle of non-fentanyl related drugs. So it gives that summary about metanidazine, as well as any case information we may have on metanidazine, and then recommendations for public health, clinicians, medical examiners and coroners, and then for laboratories as well. And really this public alert is just to make people aware that this new drug has made its way onto the drug market and into toxicological samples, and just to be aware of it and to keep an eye out if there are any adjustments that you might need to make to your scope of testing to find this. So this public alert was released in January of 2021, and it says that metanidazine is a synthetic opioid that is dissimilar to fentanyl, but related to adenidazine and isotonidazine, and was first synthesized in the 1950s. So the potency of metanidazine is similar to or greater than fentanyl as described in the public alert. And again, it was first identified in a seized drug powder in July of 2020, which is what I mentioned from the new drug monograph. So it does include that information. And then, so the demographics for the 8 cases is what we had at the time for metanidazine. So all of these cases were post-mortem, and they were all collected from August to December of 2020. And they were primarily male, as you can see in the actual alert itself, 75% male, 25% female. And the age range was between 30 and 50 years old. It also provides the other notable findings. So fentanyl was found in 6 cases, cocaine in 4 cases, and methamphetamine also in 4 cases. The last thing it gives you is the geographical distribution, and metanidazine in these 8 cases was found primarily in the Midwestern states, so Indiana, West Virginia, Illinois, Ohio, and Tennessee. So here I have the trend plots for metanidazine. This is the NPS opioid positivity in our toxicology samples at the Center for Forensic Science Research and Education, and this is by using our LCQ TOFMS. So fentanyl is not included in this just because we get so many identifications of fentanyl. It's really hard to see those non-fentanyl synthetic opioids that are also popping up. So you can see here, the one I have labeled isotonidazine, the pink line there. Isotonidazine was really that second most current wave of a non-fentanyl synthetic opioid. So it was started becoming popular in 2019, and by the last quarter of 2019, we had that peak positivity of isotonidazine. And then that wave of isotonidazine started to come down, and then we started to see a lot of brorphine. So brorphine had peak positivity in the 3rd quarter of 2020, and the wave of brorphine fell down, and metanidazine started to rise to take that place. Here I pulled out metanidazine just so we can see it a little bit better. So we started seeing metanidazine in our toxicology samples at the CFSRE in the final quarter of 2020. We did see it in a seized drug sample in quarter 3 of 2020. However, in our tox samples, we didn't start seeing it until the 4th quarter. And then we had the peak positivity of metanidazine in the 1st quarter of 2021, and that was about 1.8% of our identifications were metanidazine. So another part of the toolkit is our trend reports. So our trend reports here that included metanidazine were from the 3rd quarter of 2020 to the 2nd quarter of 2021. And the trend reports, again, include just an overview and an objective, as well as our most prevalent NPS opioid positivity, anything new, and then the most notable NPS opioid combinations. So you can see in the 3rd quarter of 2020, we have at the bottom, the new nitazine analog metanidazine was discovered, and this is from that seized drug sample in July of 2020. At this time, brorphine is still our most prevalent non-fentanyl NPS opioid with 44 identifications, with isotonidazine behind it at 12 identifications. All right, so in Q4 of 2020, you can see that brorphine, we're still seeing brorphine the most, however, from 44 identifications down to 9, and now metanidazine is on the rise. So we're starting to see that increase in metanidazine identifications. This is when we started seeing metanidazine in our toxicology samples, and we had 7 in this quarter. And then in the 1st quarter of 2021, this is when I mentioned we had our peak positivity of metanidazine. So metanidazine has now taken over brorphine. Brorphine has fallen off down to 4 identifications, with metanidazine up to 29 identifications. And then you can also see at the bottom there, it has that emergence of the new opioid nitazine analog. So all of the other nitazines we were starting to see. So you can see that brorphine and isotonidazine have really started to fall off, as well as we can see on the side the NPS opioid combinations of gives really just people an idea of what to look out for if they see metanidazine, because we saw it primarily with NPS benzos like clonazepam and fluoxetine, and then fentanyl. So here is the last trend report in the toolkit for metanidazine. And metanidazine, while it's still being seen, it's seen a lot less. We have only 9 identifications. So this is after the peak. Now we're seeing about the same number of identifications for N-pyrrolidinoadenidazine. So you can see that wave of metanidazine is starting to come to an end. Something else is coming up to take its place. So the toolkits also provide information on metabolism. So this metabolism is determined through in vivo experiments, and this is just analysis by LC-Q TOFMS, and then data processing by the metabolite pilot software. So this data has been published by Krotulski et al. In 2021. However, the toolkit does provide information on the biotransformations, formula, exact mass, and then the product ions of all the metabolites. So metanidazine was discovered to have 4 metabolites as 4-hydroxymetanidazine, 5-amino, N-diethyl, and N-diethyl. And after these metabolites were discovered, authentic samples were then analyzed for the presence of these metabolites. So the final piece of the toolkit is the piece for analytical methods. This, I believe, is an important piece because as a lot of the other reports are put out on their own, like the public alert and the trend reports, the analytical methods section is not a report that we consistently put out. This was made specifically for the toolkits. So this includes 2 extraction methods, both liquid-liquid extraction and solid phase extraction, as well as the efficiency of each. So you can see there for the liquid-liquid extraction and SPE, it gives you recovery, matrix effects, and process efficiency for both. And then it also provides 2 instrumental analysis methods using 2 different LC-MS systems. So that way, if laboratories don't have the time to be taking up their resources to make a method from scratch, they have a starting point here. So here I have just blown up the method section here from the toolkit. So on the left side, it's the instrumental method. So you have 1 in blue and 1 in green. This is for 2 different LC-MS systems. It gives you what the instrument we used was, all of the liquid chromatograph parameters, as well as all the mass spec parameters. And then it also gives you a multiple reaction monitoring transition, gives you the MRM method, and it gives you that suggested target quant ion if you need it. And it gives you that for both. And then you can see at the bottom there the chromatography of metanidazine for each. So on the right side in the bottom half of that section there is the 2 extraction methods. So it gives you detailed extraction methods as well as a suggested internal standard, and then the assessment of both, as I mentioned before, the recovery, matrix effects, and process efficiency, as well as the calculations of how we got there for each. So one important piece of the toolkits is the authentic sample analysis. So as I did mention before in the public alert, it did give information on the cases, but we didn't have information on actual reference concentration. So that's what this is here. Again, this work has been published by Krotulski et al., the same paper that the metabolism information is in. So this was a case study of 20 authentic postmortem cases. So in this paper, using these analytical methods we talked about before, it gives you reference concentration for blood and for urine. So in this set of 20 cases, the average blood concentration was 6.3 nanograms per mil with a range of less than 0.5 to 33. And then the average urine concentration was 14 nanograms per mil with an average of 0.6 to 46. It also provides the polydrug co-occurrence. So in 3 cases, there were no additional drugs besides metanidazine. 5 samples had methamphetamine. In 6, it was the only opioid. In 9, there was at least 1 additional NPS. And then primarily it was found alongside quinine and fentanyl. So there's a lot of future work to do with the NPS toolkits. So the next one on our docket is going to be the N-pyrrolidinoadenidazine toolkit, but there are a lot of other prevalent NPS that we just don't know yet. There are a lot of emerging NPS that will probably require toolkits. That way it gives laboratories that information if they don't have the time or resources to be developing them from scratch. That way we can give them that base starting point. So then I just wanted to give a bit of a years in review for NPS discovery. So since 2018, we've put out 116 monographs, 21 public alerts, 3 NPS scope recommendations, 56 trend reports, and 1 toolkit, as well as identified 197 NPS, reported 116 new NPS, and we have over 8,100 and counting total NPS detections. So I'd really like to say a big thank you to the NIJ for all their support in this project, as well as the CFSRE and NPS discovery team. And then also our collaborators at NMS Labs with the department staff and toxicologists, and our collaborators with medical examiners and coroners. Thank you so much. And my contact information is there if you have any questions. Down below, I have a QR code for our website, NPS Discovery. So feel free to visit, download, or subscribe, and follow us on Twitter. Thank you.
Thank you, Ms. Walton. And we will now begin our final live Q&A session of the day. Thanks so much to everybody who hung in there, especially my East Coast folks. So if you are in the audience and you have any questions for any of our presenters, please add them now. If any of the presenters have questions for each other, I would love to hear some of that as well. I don't know if any of you were sparked by listening to each other, but I'll start with some of the questions we already have in the queue. So let's see. Oh, I saw a good one that I was tickled by. So Arun, we saw your picture of gummy worms on your slide, and we were just wondering, why was the name earthworm chosen?
I can answer it. Yeah, so basically we needed a way to delineate between our samples, and when we were worried with numbers, that it would be like an expectation that, oh, after version 1 you've got to have version 2, you've got to have version 3, and so we just went with letters. And then we chose insects because cicadas were really loud when we made this decision at version 3. Yeah. And then we've just followed along with insects. So if you have any recommendation of insect names, because we're already running a little bit thin, like we got firefly for F coming up next. I don't know after that. Grasshopper. So yeah. So no, it's just kind of for fun, nothing special.
I love it. That's how the naming convention on my pets started, and now I'm similarly stuck, and I have to limit my pet supply, I guess. Let's see, what else do we have in here? All right, so Sarah, how many laboratories and other reporting agencies provide that emerging NPS trend data? That's a very popular question. How much goes into that, and how much of it, I know some of it is tox and some of it is seized drugs, but do you have sort of a sense of what the breakdown there is?
Yeah, it's a good question. So to answer your second question, it is primarily biological samples that we test, but we do have some seized drug samples. It just depends what we get at the time, but normally it is primarily just biological samples. And then for your first question, so our lab does provide all of the data for NPS discovery. So we run the samples, but there are we have a lot of collaborators that send us samples to run, like medical examiners and coroners. Our other collaborative labs, they send us these samples to help us provide that data for everyone. There are other early warning systems that do pull data from other labs, but we are kind of separate from that.
Thank you. I have a question that actually I think is applicable to Glenn and Robbie. I'd like both of you to address. How do you anticipate that your approaches would be incorporated into a traditional forensic lab workflow?
Glenn, are you going first or am I?
You can go first, that's fine.
All right, sounds good. So what we're doing is setting up our system so that it essentially is software. So we're building a graphical user interface that makes it so that once it's installed on a person's computer, all they have to do is upload the mass spectral data. In this particular case, it would be DART MS data, but also they could upload infrared data. And when they do that, then the process would simply be automated, and they would simply need to go through a series of clicking exercises to get two types of information. One of them would be, for example, for tryptamine, but we're doing this for 4 classes of compounds: cannabinoids, opioids, cathinones, and so on. And tryptamines, of course. And so the idea would simply be, if they've got some substance, some white powder, and they're able to get a DART mass spectrum of it, and they're able to acquire this at voltages where you could get fragmentation, and they're also able to get an infrared spectrum, they can simply upload both of those, and the software would then give them candidate structures for what the unknown could be. So in terms of workflow, the idea would be that they would do the traditional GCMS. And when it's discovered that the compound is new, in the sense that there isn't a retention time match, or there's a retention time match, but there's no mass spectral match, that type of thing, then they would be prompted to simply take the same data that they may have generated by DART and MS or infrared and simply upload it into this graphical user interface and get a series of candidate structures, because that would be your new psychoactive substances, presumably. So briefly, that's what the idea would be.
Thank you. That was very clear. Glenn, your turn.
Yeah, so I was at AAFS last week and gave a really similar presentation there, and I was talking to a crime lab director afterwards, and I asked him, if we were to make this available to your practitioners, how would they want to use it? I said, should we develop an Excel spreadsheet? They just put their data in, and he was like, maybe is there another way you could I'm like, and he's like, yeah, no, you don't want people to get lost in Excel. So he said some people just are not very good in there, and get turned upside down and stuff. So he recommended that simple software, a simple graphical user interface that's pretty much easy to use. Just import a spectrum and give me the answer, and remove as much functionality as possible. So that will be our plan when it comes time to commercialize or distribute or try and make this practical for practitioners.
Thank you. Ed and Arun, obviously you are already at the published point of the tool, and one of the things that you had incorporated, because you had that great end user feedback loop incorporated into your project design. So you incorporated the enhanced reporting capabilities, but I know that everybody recorded a month ago, and I know how fast things move. So are there other QA or sample tracking capabilities or anything else that are included in the most recent release that you may not have had the chance to mention in the talk?
So version 1.1 should be out next week, but we're trying to finish up version 2 right now. In terms of sample tracking and QA/QC, there's not a whole lot new in there. We still have the same quality assurance things where you can't upload multiple files of the same name. Your data files are going to be all spelled out. What we're currently working on now for version 3, I guess, is a SQL database where you can actually store and keep a history of all the searches that you have done. We were thinking of it less from QA, more from data mining perspective, but it would definitely be another QA type tool that we would have in place.
Yeah. And just to kind of go back to your second question that you asked, Glenn and Robbie, just from our perspective as people that have recently done software, it's crazy how quickly the software becomes not simple, especially as you interact with the labs and you start seeing, because that's kind of the process that we took, where we were interacting, trying to figure out what they wanted in their software versus what we thought they wanted, which are two not necessarily the same thing. And so, it's nice with the NIJ to have that pipeline of interacting with practitioners. And so hopefully when you guys are developing your software, so you can follow a similar kind of pathway.
I just want to say he was not incentivized to say that, but I think he's done a really important point, because that we see that so much, where if you don't have that interaction with the crime lab directors, with the benchtop chemists, like you said, you can have a very clear vision of this is the solution that you will want, and then you may miss the mark entirely because of something that you're not aware of, that isn't really relevant to your life in academia or in sort of the pure research. Sarah, so that toolkit is extremely comprehensive, obviously. how long did it take to compile all of that and make it available? And as a follow-up, what made you decide, CFSRE, decide to go that sort of full toolkit route?
So it does take a very long time. I would say it probably takes between like 6 and 9 months to fully put together a toolkit. And the nice thing about the toolkits is that a lot of its pieces are out as it goes. So every time we see a new substance, a monograph goes out. Every quarter, our trend reports go out. So a lot of that information is already available. The problem that we as the center kind of saw is that people have to go to all these different places to pull all these reports, and it would be nice to have one central thing to pull everything together, and then also give people extra information, like information on metabolism and other analytical data that at the time of the drug monograph we just don't have. So once we have a lot more samples and more cases, it's easier to get more information and then put that out to the public.
Got it. Thank you. Robbie, as you know, Sarah's talking about the rise of these, bromine coming up and going away and metanidazine coming up. So how would that be impacted or impact on your process? And so when a previously unknown NPS, as you say, comes up in whatever context, does it need to undergo this process, the HRMS, the hierarchical clustering, to determine that potential drug class, or is that more of a, are there other methods that would be more appropriate for sort of that initial classification, and then this would be once we're getting into all of these tentacles of isomers?
Yeah. So the whole idea with this project and what we're doing is that we want it on the front end, as close to the front end as possible, as in once you reach the point where you know that this is brand new, to have a mechanism by which you can screen your compound. So there are two tiers of discrimination. The first tier is where I didn't talk about this particular element of the work, but the first tier is where it tells you this is a cathinone, this is a cannabinoid, this is a tryptamine, this is an opioid, right? So that's one tier. And again, this would be automated. And so the first thing you would do once you realize you have something new is to screen it against this user interface, this software. Then once it gives you that classification of what it is, so for example, a tryptamine, then there are two tiers of discrimination. You have a tryptamine, but as you know, it can be a methylene dioxy derivative, it can be a pyrrolidine derivative. So it'll tell you that I'm 98.2% sure that this is a methylene dioxy derivative. And then, so you go in knowing that, which is something that you wouldn't have had just flying blind. And then the next tier would be where it gives you candidate structures. So the idea would be that once you reach that stage, the candidate structures are whittled down from, say, 1,000 to 4. And the 4 are typically isomers of one another. And so at that stage, it becomes fairly straightforward for you to then say, can infrared help me to figure this out, for example, based on certain features that I see maybe in the fingerprint region and that kind of thing. So that's how it would work. And that's where you would then want to fall back to other procedures, other analytical tools that you have in your lab that can help you. But you're no longer flying blind, so to speak, because you've got not only information on what class of molecule it is, but what class within a class it is, if it happens to be an unknown. And that process, by the way, once you have your data, really would take you less than 15 minutes, from start to finish in terms of figuring this out. So it would be really rapid.
That's great. Thank you for that. There's a question from the audience for you again. Will the DART MS work with mixtures? Mixtures are always the question.
Mixtures are always the question. So the short answer to the question is yes, but that would be facilitated actually by a GC experiment. So the idea is that, when the typical procedures would be followed for a drug that's coming in, right? So the gold standard is GCMS or GC. And so you would start with GC, and that would tell you that you've got this mixture. Now the algorithms that are associated with this, particularly if you perform the DART experiment under collision-induced dissociation conditions, it's complex. But what we've done is we've set it up so that the algorithm learns to recognize patterns within very complex collision-induced dissociation spectra so that it can extract out those peaks that tend to be associated with a particular class of molecule. And so that's what it's designed to do. So the short answer is it will work for mixtures, but it's certainly helpful to know on the front end, once you've done a GC experiment, that you're looking at a mixture.
And
one more thing I would say about that. The DART experiment is first done under soft ionization conditions. And so because you're looking at the protonated precursor, you're seeing a peak for every different molecule in your system. So that actually confirms for you that you have a mixture. Sorry for the interruption.
No, that's a good point. And then I was going to ask Ed Naroon, how does the data interpretation tool deal with mixtures?
Yeah, so the underlying algorithm in the data interpretation tool is what we call the ILSA, which is the inverted library search algorithm. And actually, just to Robbie's point, it's looking for substructures in the ISCID mass spectrum that match pure compound spectra. And so we've shown that works really well for simple mixtures that we create in the lab. And we've started evaluating it with real seized evidence. And it's working okay. It's harder to tell if it's, how it expands. If you have a mixture of 10 drugs, at different concentrations, it might be harder to pick up the low concentration drugs, but at least the main components of the mixture, we are able to identify just using the inverted library search. And so, yeah, of course, again, it's only with the limited test cases that we've used. And so we don't want to say anything universal, but there's promise here.
Great. Yes, we certainly will not hold you to the magic bullet. Glenn, how many data points or samples do you need in a training set to appropriately produce those mean residuals with the minimal false positive, false negatives to set that threshold?
Yeah, so that's a really good question. And basically, there's no one answer. It depends on the variability of the instrument, on the variability of the sensitivity of the drug to that particular instrument, and also the variance you get for that drug between instruments and between vendors. We have looked at, in terms of size of database, we've looked at building models based on 10, 20, 50, 100, and 150, and thousands of spectra. And generally, once you get to more than like 70 spectra, the models start not being significantly different from one another. So there's kind of a plateauing effect. When you get so many data, it just adding another one doesn't make any difference. But of course, when you have only 20 spectra, then sampling statistics dictate that you get some bias one way or another. So that's one thing. And then the number of mass M/Z channels to use within each model is also, there's not really one answer. Some drugs have more unique spectra than other drugs. You might only need 3 or 4 ions to be able to discriminate that particular drug from all other drugs. Other drugs like cocaine and its diastereomers are much harder to tell apart, and so you may need more ions to tell them apart. So there's no one answer. We use 20 right now because we can guarantee that any drug will give you 20 peaks if there's a remotely reasonable concentration of it, even amphetamine and methamphetamine, whereas and it will also give you reasonable limits of detection because 20 peaks, even if something like cocaine would give you 100, some of those low abundance peaks would drop out very quickly once you start reaching the limits of detection. So right now we do 20 peaks and more than 50 spectra. I guess that was a short answer.
It is. Sorry, my puzzled face is because we have a question from the audience that I'm not sure who it's meant to go to based on it. So I'm just going to throw it out there, and whoever wants to answer it can. Can the software predict the mechanism of toxicity? Yeah, all the drug chemists are being quiet, and Sarah's like, please don't call on me. That's not right. So I'm not sure, maybe the audience member who had the question can clarify that in the chat. We're almost to the end of our Q&A, but if they do clarify who they were intending that for, we can circle back to that. So do any of you have questions for each other before I kind of go to one of these last questions that we do have in the queue? All right, nobody wants to put anybody else on the spot. So Robbie, as it stands right now, with the combination of the DART high-res mass spec, soft ionization, the 90-volt fragmentation, is sufficient to identify and distinguish most cathinone or tryptamine isomers?
The short answer is that the what we have so far actually can be used as a screening tool for known compounds. So if it turns out that the compound is known and happens to be part of the database that we've built so far, and of course, it's continuously growing, the answer to that question is yes. That's the short answer. However, the reason why we coupled infrared with the DART mass spectral data is so that we can have an edge in dealing with isomers. Because, you know, when you, as I think most people here on this call know, it's typically what happens is you can have structural variants where you simply change you can have positional isomers and they still retain their psychoactivity. And oftentimes by mass spectrometry, it's not possible looking at isomers that differ in terms of positions around the benzene ring to distinguish between them. But that's where infrared can really help you. And so the short answer is that the current form of our system, where it incorporates within it infrared spectral data, the short answer would be absolutely yes, because that's why we did that. The DART data by itself will get you only so far if you're dealing with positional isomers. So it's really great that we were able to fuse that data with infrared data, and we were surprised at just how robust that system is. Works very well.
Perfect. All right, well, we have come to the end of and run over a little bit on our Q&A, so I thank you all for being here. If anybody in the audience has any further questions, everyone gave their contact information, and I know all of these presenters are eager to talk about their research. So Lauren, you can take us out. I'd like to thank all of our presenters again, as well as all of the audience members for joining us on the first day of NIJ's 2022 Forensic Science Research and Development Symposium. Please tune in tomorrow at 10 a.m. Eastern to hear presentations and discussion on forensic biology, DNA, anthropology, and pathology. Have a great evening.